The Argus Institute for Veterinary Disease Control in Mödling is one of Austria's most important and modern institutions in the field of animal health. Its experts have been focused on one objective for more than 100 years: the diagnosis of animal diseases and measures to maintain animal health based on state-of-the-art knowledge. The Institute for Veterinary Disease Control Mödling examines animal carcasses, blood and organ samples, as well as animal products for potential causes of diseases, in particular for microbial pathogens. Its special focus is on diseases that have a considerable economic impact or which can be dangerous to humans. Such diseases are called zoonoses. Examples of zoonotic diseases are rabies, influenza such as bird flu, West Nile fever, tuberculosis, salmonellosis, and many more. Foot and mouth disease or blue tongue disease in herds can cause considerable economic damage. Thus, quick diagnosis and veterinary health measures are essential. The testing of animal samples suspected by the official veterinarian to carry notifiable diseases is analyzed with priority, and the regional responsible authorities and the Federal Ministry of Health must be informed about the test results immediately. In case of positive findings, the appropriate epidemic control measures are carried out instantly. International animal trade agreements are increasingly bound to certificates stating that the animals traded are free of any pathogens. Such export tests are designed to minimize the spread of disease as much as possible. In addition to tests commissioned by the authorities, the Institute also serves as a contact point for local veterinarians who submit their samples as part of private tests to expand their diagnosis based on the results of modern laboratory methods. Animal blood, organs and samples of the animal environment are documented and forwarded to the relevant departments, where the tests are carried out and test reports are written. Animal carcasses are brought to pathological examination. Following an initial evaluation, samples are taken during necropsy for further diagnostic tests. More often than not, these further tests to find out the cause of the disease have to be done. These include isolation tests, for example, during which a special nutrient solution is added to the sample, causing the infective agents to multiply, or in the case of viruses, an infective tissue suspension is inoculated in receptive cell cultures, or into embryonated chicken eggs. The bacterial growth or virus assembly usually results in clear identification. Electron microscopy is used in certain cases, as it can reveal individual viral and bacterial agents, especially new or re-emerging viral pathogens and cellular host-pathogen interactions. Molecular biology has become increasingly prevalent as an alternative to rather time-consuming culture and enrichment tests. The polymerase chain reaction, or PCR, is used to identify all species of viruses, bacteria, fungi or parasites on basis of their nucleic acid sequences. It can be used for genotyping individual gene variants within a species, the principle in identifying the outbreak of a disease and in finding out more about the pathogenicity and infectivity of a certain agent causing the disease or outbreak. However, pathogens are not always present at the time of sampling, for example in the case of overcome infections. Nevertheless, proof of these infections is relevant for epidemiological reasons. For that, serological diagnostics becomes invaluable. Animal blood samples are tested for the presence of antibodies against infective agents produced during the immune reaction. BSC tests are a special case. Before the meat of a cow above a certain age limit will go on sale, a brain sample of the animal must be tested for infective prions. Time is of essence here because the carcass may only be processed following a negative test. The Argus experts at the Division for Animal Health have one common goal: ensuring fewer animals get sick. The close and reasonable interaction of all Argus divisions and the intensive cooperation with international organizations, as well as with Austrian authorities, is one of the Institute's strengths and makes it an important partner in Europe.